In this video, we'll learn about the process of gene expression. Gene expression takes place in two stages. The first stage is formation of single-stranded mRNA from a double-stranded DNA, which is known as transcription. The second stage is formation of proteins from the single-stranded mRNA, which is known as translation. These two processes occur at different sites within the cell. The first stage, which is the formation of mRNA, takes place in the nucleus. Once the mRNA is formed, it travels to the cytoplasm of the cell where the translation process takes place. So here we'll see how the transcription process occurs. To know this process, first we should know which all biomolecules are required to carry out this process. The biomolecules required are positive transcription factor, which attaches at the regulatory region to carry out the transcription process or to initiate the process. The second biomolecule is RNA polymerase. RNA polymerase helps in attachment of the new strands or the new nucleotides in order to form an mRNA. Ribosomes are required for the translation of mRNA to proteins. mRNA destroyers. These are required to digest the mRNA formed in the cytoplasm. Negative transcription factor. Negative transcription factor helps to stop the transcription process. The process occurs in few stages. The first stage, the positive transcription factor attaches at the regulatory region. After the attachment of the positive transcription factor, the RNA polymerase attaches and starts the transcription of the transcribed region and forms a single-stranded mRNA. The single-stranded mRNA travels from the nucleus to the cytoplasm. In the cytoplasm, the ribosome attaches to the mRNA, which translates the information of the mRNA to form a protein. This mRNA then needs to be destroyed. So a protein present in the cell, which is known as the mRNA destroyer, attaches the single-stranded mRNA and digests it so that it won't be functional anymore. In order to stop this translation process, the regulatory site needs to detach the positive transcription factor so that the place is available for the negative transcription factor to attach and stops the further transcription process.